I thought that Robin's thing was great. Have we recorded that? Because I want to get, I mean, I know that story's been out there, but I just really appreciate hearing it. And I
All
can't
right.
imagine.
Hey guys, welcome to the heart of New York City, right here at our little studio, nestled on 38th Street between 5th and 6th Avenue. We're talking about regenerative medicine, and we got some great people here, part of this live stream that I think you're really going to want to tune in for and listen and learn. I got my cousin Matt. Matt, why don't you take it away and introduce some of these people and see what we're kicking off?
Sure, sure. I'm totally excited because we're here tonight talking about regenerative medicine for dummies. That's kind of the framework, but it's more like a cocktail party where we could just, have the conversation go wherever it goes, which I'm fine with. And who we have here today, we've got Mark, who's at the bottom of the screen. He's a biomedical engineer and is an amazing guy. He's out like making his way with a company that tests drugs, and he's really specific, I'll tell you. Sangeeta is at the top here. That she is a grad student, PhD at the University of Cincinnati. We have Elad Katz, who is a complex biologist, and he is calling us from England. And it's 1 o'clock in the morning, and he's a trooper to join this. I'm so glad that you're here. We have Robin Farmian. Did I pronounce it correctly, Robin? Good. All right. She is a world-class speaker and speaks all around the globe, and just finished up a speaking arrangement at I think about 2 hours ago. So she's joining us now. And on the bottom of the screen, I'm not sure who is here. I think it might be Ira Kunze in England. Is that correct?
I don't know who you're talking about. You talking about me?
Yes. Who are you?
I am Irma Rastigaiva, and I'm joining from Boston.
Oh, that's so great. How are you doing?
I'm doing great. I'm doing great.
Fantastic. And so, yeah, we're just throwing around a conversation about regenerative medicine. It's kind of regenerative medicine for dummies because Pat's here. And so, no, really, we've got a feed that just goes to your average non-scientist person, and each of your respective fields, it's like impactful. Absolutely. So, Robin, could you just tell us again about your misdiagnosis early? Because I'd like everybody to capture that.
Sure. So I'm Robin Farmian. I have a book called The Patient as CEO. In addition to all the technology that it talks about my backstory of being misdiagnosed at the age of 16 with an autoimmune disease, having 43 hospitalizations, 6 major surgeries, and 3 organs removed for that. At the age of 26, this is 7 years after I'd had my entire large intestine taken out. My doctors were telling me I was cured, but I was in extreme pain. So they had me on 80 milligrams a day of methadone, which is massive opiate dose. And told me that was essentially going to be the rest of my life. And I said, no way, I'm taking control of my healthcare. Fired my entire team, replaced it, ended up getting diagnosed correctly, put on Remicade, which is an IV medication I get every 6 weeks. And I literally went into remission overnight. So that's my main message is really how patients should be taking control of their healthcare, because it can make literally a life or death decision, right? It can change the game completely.
Awesome.
Yeah.
And I just want to do a little footnote. When I was a kid, I was operated on by Dr. C. Everett Koop, who was the Surgeon General at the time.
Yeah.
And he took, I think it was 6 feet of my intestines out.
Wow.
When I was like a year and a half old. I didn't even know that you could have that, much intestine at that time. But that's pretty wild, Robin. Great story. Matt, back to my cousin Matt.
Yeah. Well, I'm very excited to hear about the patient kind of reaching out. I think about it now. I mean, well, I'll get into it, but I'd love to hear from Sangeeta just what you're doing as far as your PhD, what you studied, and, find out a little bit about you.
Yeah, so my name is Sangeeta Kandoi, but people call me Sangs. I did my master's in India, and I was working in the stem cell industry in India, which basically deals with hematopoietic stem cell banking and mesenchymal stem cell banking. We also had a lot of patients who had enrolled for thalassemia and got treated with the stem cells which we had stored in a bank. So I was working there for almost like 8 years, and then somehow something pushed me that, you know, I should do my further studies and do my PhD. And that's why I got a lot of support from my management to do this PhD. And I got enrolled in a university in India itself, which is in collaboration with my industry. I did my basic coursework qualifying in India, and then I had to do my project. And then there was a debate whether I should do it in India or United States. So then everybody supported me, and they wanted me to do it, you know, on a big scale, whatever it is they call it. And then they sent me to University of. They initially sent me to Loyola University, Chicago. So I started my project. I started working on induced pluripotent stem cells with perspective to cardiovascular disease. So,
Excellent.
I mean, something specific to tell it is hypertrophic cardiomyopathy. So, I mean, hypertrophic cardiomyopathy, and one specific mutation which causes hypertrophic cardiomyopathy is 25 base pair deletion mutation. And that's like very common in Asian population, and being an Asian, I got very instigated, or, you know, that, you know, I should work on this disease, or I should at least do something novel, you know, so that when I go back, I carry something.
So you're just like Robin, you're very passionate, and it's very
very
home.
passionate.
That's awesome. Who else we got, Matt? Okay, Matt's sound.
Matt's on mute.
Matt, you're on mute. See?
Sorry, I said we had Elad over here in the
Regenerative medicine
corner.
for dummies.
He is killing it. He's killing it. In England, it's 1 o'clock in the morning. So Elad, please tell us a little bit about your work.
Yeah, so first, I'm based in Scotland, not in England. I know it's far from you, but so I am. My area of interest for many years now has been really constructing disease models from regenerative medicine, but from in other ways too, to really understand how to discover better drugs. Our thesis has always been that if we discover our drugs using things that look more like the disease that is in the patient, we'll stop wasting a huge amount of money in the drug discovery stages. Because what happens today, we do this on a huge scale using things that don't look anything like the disease. And then we get to phase 3, we pay up to $1 billion, and oops, it doesn't work. And that's a big problem. It's been really hard to break the habits of big pharma, because they're so used to spending the money at the end. So it pays Mark's salary very nicely, but it doesn't help people like Robin very often. So that's been something I've been working on in different diseases. I personally are quite a regenerative medicine skeptic. I don't think that that's going to solve all of our problems today. I think that certain things there are amazing science, but not necessarily translating as much as the hype is to being applicable necessarily to making better drugs in general, better precision medicine, if you like. But I think that we need to have the aim of developing things as quickly as possible and as accurately as possible, and that's really what matters. If something is sexy or not is beside the point. The point is that we need to make the whole process more efficient. And one of the things that I've learned, which may be news to most people, or certainly news for me over the past couple of years, is really how much of the medicine discovery and development is in the hands of chemists. And I would say I was trained as a chemist, so I can speak badly of them. There's very little biology input. There's let alone very little clinical input. And this is where this clash of civilizations happens. A chemist can make fantastic things that look like drugs, but they might be completely outside of the biological context, and that's why we're spending so much money. And a lot of the drugs that we make, just like in Robin's story, all of a sudden, completely by accident, we might discover that they are fantastic in a completely different disease that we never really try to understand. So we have millions of patients worldwide that we can help tomorrow with things that we already have. Of course, drug companies, if it's in patent, are all for it. But the moment it's over the patent, they couldn't care less. And Remicade is one of these that is falling off there. No pharmaceutical company would now fund a new clinical trial into drugs like this because it's going to be expensive, it's going to be risky. And we need to figure out how we decide which diseases to even try and who on earth is going to pay for it.
You know, that's wonderful. And that brings up an interesting conversation that we had recently with a friend of ours, David Goebel, who helped to fund Organovo. Have you heard of Organovo, Elad?
Yes, I know them quite well.
Oh, wonderful. Okay, so David was talking about the Methuselah Foundation, which he started, and he is creating scientific setup studies and willing to fail, like absolutely from the beginning, ready to fail. And if they hit it at 80% of what the goal is and they still fail the goal, they still have tremendous success because of that very thing where science is often dictated by shareholders and large corporations where they're looking for easy wins, and they have to show and publish studies. They publish studies that are only wins. And the more hits that they get, they've got to follow the wins. They don't go after the riskier pathway. So I'm glad that you're very familiar with Organovo and some of the disease tissues that they can create that are actually, I think, model human tissues.
The important message is that you have to use those failures. So what happens at the moment is that you fail a lot at the very beginning. Using those funny models that don't really matter. And then as the money piles up, there's more and more pressure to push things forward. But what I personally find always bizarre is the mindset that if something, and that has changed a bit with immuno-oncology because they decided that you can't predict it. That's another argument, but maybe it will lead to a bigger shift in general. You see that if something failed in phase 3, instead of cutting it in a different way and say which part of it succeeded and can we build on that, often the whole thing is taken off the table and we start again as if it never happened. It's a very, very strange way of using your money. And I think that, again, all the way to the patient, there has to be pressure on, hold on a second, it's not that the drug was not safe. It may have not worked. The failure was that it didn't work as the way that possibly your marketing guys really wanted it to work. If we could have defined a smaller portion of patients that this has worked for, that's still a win. So it's redefining what the win is. Stop looking for the failures because, as you said, they want to be safe. So if it's not 100% what they predicted, they just throw their hands in the air and say, this is it.
Yeah,
one of my favorite quotes of all time is, "You can't steal second with your foot on first." So
that's nice.
Let's
Great quote.
Let's finish.
Yogi Berra.
Yogi Berra. Oh man.
So I have a question. All right.
What's that?
So we were talking a little bit with, how do you say, Sands? About iPSC cells. I heard her say those, which are induced induced
Pluripotent.
pluripotent stem cells. So for me and kind of Robin's story, not quite like Robin's story, but other people who have to read her book and become a patient as CEO, I think that iPSCs may hold a tremendous benefit in a whole host of areas because of the no rejection. These are cells, tell me if I'm right, that the Japanese scientist was able to turn from just either your fat cells or your skin cells, can turn these cells into any cells in your body.
Yeah,
Go
I would
ahead.
like to add the IPC, what I made from the patients were from the patient's urine. So it's only from the urine. The pee, what they do, I collected the pee and then I extracted the pee cells from that.
Wow.
And then I converted that P cells into iPSCs and then differentiated those iPSCs into cardiomyocytes. So it was like totally non-invasive procedure.
Yes.
There was no pain for the patient. There was no ethical issues involved. So it was very straightforward, but definitely it's a very labor-intensive procedure. I do agree with that.
Yeah, and depending on what you create with the iPSC cells, and you're very specific to what your study was looking at, but
Right.
it seems that we could use these cells for all kinds of things. You can train eye cells, your muscle
So
cells,
it can
cardiac.
get differentiated into 220 different cell types, which we have in our body.
Pat, are
So
you
it's
listening?
very flexible.
You got to be listening, buddy. 220 different cell types. You know that cell type.
I'm hearing you. I'm also tuning into like Facebook Live. We got a lot of people lining up for questions. I say we finish it going around the horn. Mark.
Okay.
Let's hear from Mark, and then let's get into a whole,
bam,
fire
powwow,
moving around.
Yeah, let's go. Let's go.
I love it. Mark, what's happening, brother?
So I was just going to comment off of this. The timing of what's going on now. We started to mention just a little bit about precision medicine, and I personally like to go against that grain and say it's not so much the precision, because in the clinical trials that I deal with, there's a lot of differentiation for accuracy and precision, making sure we're measuring the same thing over and over again, and also the same thing, whether it's at Stanford or at Yale or here in New York or in Tokyo. That doesn't happen necessarily when it's the regular doctor that you see every day. And when they send you out for a regular EKG or an ultrasound, they take that number and they think it's gold. They don't know what's necessarily lying underneath. So I would say it's the personalization of medicine, which then leads me to everything that we had just started to touch on. The time now, the patient centricity, the ownership of data, price transparency. There's a lot of stuff that, it's not pure technology like digital health that's going on, but technology influencing, organs on a chip, Organovo, even from my imaging standpoint and what we were talking about with better early preclinical before taking the drugs into humans. It used to be in rats and mice studies, you'd give the animals the medication and you'd have to sacrifice them and open them up and take a look. I can't tell you how many, even in the past year or so, people that I would have to say, the imaging technique that I use in humans, I can apply it to your animals so that you don't have to sacrifice them. It saves the animal pain, money, time. I mean, these type of things, it really drives back to thinking entirely different. So the culture aspect, I'm very pleased to see everybody's different perspective, but it still ties back down to how can we change it so it's better for us as the consumer of health, medicine, etc.
I love it. Now, one of the things that, just to throw some little, get my technical thing up, just to throw something out there for the crowd, because I'm not sure how many people, there's a lot of people online right now listening in live, and just throwing out a little something that I read earlier. It was a guy at the Mayo Institute. He was talking about regenerative medicine, and he was talking about how they would do hip surgeries. And he's saying that the stem cell treatments, it halts 80% the progression in these, I guess this hip, you know, whatever it is, the hip issues that they're having. So and it postpones it for like 2 to 5 years. So, I mean, is there any stories out there that you guys can just say, hey, this is what this science is doing, just because sometimes you need those broad strokes that just say, wow, like to me, you're going to go in for a hip surgery, but you're able to postpone it for 2 to 5 years. That's incredible to me. Absolutely incredible. So, I mean, Robin, you know, your firsthand, you know, you like your story is incredible, but any other like just quick stories we can go around the horn and just figure out and just say, hey, I witnessed this or I witnessed that, because then I think that would be a big wake up call for some people to say, hey, this is something I need to know more about. And then, you know, to kind of continue, just to ask you guys, how long has this been around? How long has this science been around? It's not new, right? Has it been around like 15 years, something like that? And then just kind of, you know.
So stem cell research has been there for almost like 5 decades, but then the clinical research has been picked up over the last 2 decades.
Wow.
Yeah.
And now the last, and then just recently, I think it's probably made some huge advancements, correct?
Well,
yeah, right.
I would add to your example the hip and then the knee for cartilage regeneration, osteoarthritis, rheumatoid arthritis, etc. When you're looking at implants and other things, being able to just do, you know, a little bit of an injection with some of these to help the natural biological process rather than just the hardware sawing, cutting of the bone, putting in an implant, the bone metal interface, and is that going to degrade after the implant's in? All of those, you know, truly medical concerns and questions that you may have down the line can help be limited by some stem cell treatment. That's a big difference.
Yeah, and not only that, but they're walking into their mom and pop doctor's place, and this is what gets into what you were talking about, which is like, how do we get that down to the consumer? And how Robin did it was she took control of looking for it and she found it, which is great. But a lot of people will just kind of shuffle into the, you know, orthopedic office and sign up for a surgery when maybe they could have benefited from some stem cells in the right spot. I'm not, you know, it looks like some stuff is definitely, they're looking at stroke patients with ischemic stroke. They're looking at, you know, macular degeneration. These are multiple areas where potentially stem cells could make a tremendous benefit. And right now, I think there's a little bit of a battle in Texas right now. So like if you wanted, let's say, okay, I want to go. I've got to have a hip replacement, but I'd rather try a stem cell therapy first as my own patient as CEO move. Well, what am I got to do? I'm going to find a bank that's got the stem cells or somebody that would create them from me. But then I think people had to go to like another country to get therapy. Texas was banking them, but they weren't allowing the procedures. Now I'm not part of a stem cell company at all. I'm just like a person that's interested. And if, you know, my mom had to have a knee replacement, then, you know, I'd have to look. And I think there's some legislation that's in Texas now that you can actually, that may have passed. I got to like check the facts, but I think it may have passed that you can get some therapies done. Now, even when I'm talking about this, I know there's some people that are into aesthetics that are looking at, oh, I want to have better looking skin, you know, so they're going to use stem cells for that. And it's the fountain of youth. I'm not talking about that. If everybody's online, all that, I'm talking about like really, if you've got a surgery, and they've done it in open heart surgeries where they've injected stem cells in, and I believe the anti-inflammatory effect of the stem cells, and at the same time the healing component these patients are studied and they're doing fantastic afterwards. So I don't know. What do you think about that, Elad? I haven't heard from you.
Well, I think that on one hand, it's great to see the anecdotal evidence that, you know, in heart surgery or in knee surgery, we can get benefits from stem cells because the technologies certainly exist at clinical grade to isolate even on the spot. There's a German company called Miltenid, for example, that has a self-contained machine that you can wheel in.
What's up, John?
Plug the patient in, isolate the stem cells from their blood, and inject it back into the patient on the spot, right? So it's fantastic stuff. But all we have there is at the moment anecdotal, and I personally believe because there is no big money to be had there. So it's not that something truly prevents us from doing a proper clinical trial on, say, heart patients and see that. Maybe where things could come along, again, I know better the European scene. There are biomaterials companies that would, for example, patch the heart with stem cells, but their interest is actually in the biomaterial. So they might have a monetary incentive to actually do this because they sell the patch rather than sell the stem cells, because the stem cells, if all is good, are actually yours. So that's one level. And I think that when we're talking about acute injury, I think that we are well ahead to some degree because we are actually building on decades of things like bone marrow transplantation. It's not actually, at its heart, it's not actually that different. And I think that's fantastic. Where it's much, much more challenging is where we're trying to start looking at the more advanced things. So on one hand, things like CAR T, which is a way to reprogram the patient's cells, that's not stem cells, into fighting one's cancer. That's been going okay with blood cancers, much less so with solid tumors, lung or breast or whatever. Despite the rumors, we are nowhere near treating anybody with these kind of things, despite big companies such as Novartis spending untold amounts of money investigating into it. And the other thing is really replacements. This is where I know Matt loves Organovo, but I think that we are at least a decade away from implanting any stem cell derived tissue into. Well, and there's issues of scale, but there's an issue here that the stem cell people don't like to talk about, is that the iPSC cells are actually kind of more like an embryo organ. They're not really like an adult organ. So if you test on the nice little mini hearts that you can make from stem cells, you're really looking at something that is much more reminiscent of embryonic heart, which is great because we'll never test anything on children. But actually, you probably wouldn't put that kind of a heart into a person, assuming that you can grow it to scale, or liver, or kidney. Yeah. So we are facing, there's another technological leap ahead of us that what upsets me personally as someone that likes modeling is that we don't talk about it. And if we don't talk about it, we wouldn't solve the problem.
Well, it's funny you talked about, like pediatrics and small, and you talked about CAR T. I was reading about the young girl. She was like, 3 months old or something. I think it was in 2015, and they cured her of leukemia with gene editing. So it was genetically modified cells that were given to her. It took care of the leukemia, which blows my mind. I can't imagine being a parent of a child, and again, patient as CEO, what can be done? I'm at the end of my rope. What can be done? And then finding this therapy, which is gene editing, which is, now we got stem cells, now we're into gene editing. All right. So, but you've got it together and changing a family's life. Yes.
To add on to this, I would like to share one of the live story which has happened. I mean, I was a part of that team. So there was this girl, a 7-year-old girl, who was diagnosed of thalassemia, and she belonged to a very poor family. So the family didn't have money even to have, the blood
Right.
transfusion every month. So
She was. You said she was diagnosed with thalassemia.
thalassemia.
What's the prognosis for that? I don't know that disease.
So thalassemia is wherein you have defect in your red blood cell. So it's a kind of red blood cell cancer. Leukemia is a kind of white blood cell cancer. So we have like red blood cells and white blood cells in our body. So leukemia goes into a part of this white blood cell cancer, and thalassemia is something. It's a red blood cell cancer wherein the hemoglobin is not being transported properly, and that's why you don't have a proper oxygen supply. So I can share the link of that particular link, the success story of that, and you can all have. It's just a 11 minutes video which we had taken with that entire family to show how that girl has come out of leukemia, thalassemia, by the stem cell transplant which we did.
Nice.
Yeah.
That's so great. You know what? It's so much about, as this conversation gets larger and larger and more people get interested in these sciences and start putting money up, and the field just even gets more robust than it is. It just gives people hope because, again, I've mentioned it before on other live casts. Matt, who's on the cast here, is my cousin, and both of our mothers passed away from Alzheimer's. So it was always like he and I mean, I don't give up, but what was always to be said was like, hey, this happens and there's nothing you can do. 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 So this kind of gives me hope, and I'm like, oh man, I wish my mom could have hung around for another couple of years. Maybe this could have helped her out, or maybe it can help some other people out. And that's why I'm so passionate about having these conversations, because the more I learn about it, the more hope that I get for other people. And I think that's just a wonderful thing. And I applaud the work that you guys are doing, and it's awesome. Mark, do you have a story that'll just like perk somebody up that you've been involved with?
No, not just yet, but I was actually just going to say, because again, this is kind of tying together the anecdotal stories, the technology piece. It's like the patient CEO. It's the early adoption. You go back and look, how long has this been around? Some of the stem cell therapies you were talking about, legislation. Think about the professional athletes, the early adopters who would not be able to get some of the treatments in the U.S., going out to Europe, getting what they needed done, whether it's some of the examples, knee and arthritis and things like that, to now, in this day, getting educated, and not just through their normal channel of the doctor, and not just Dr. Google, so to speak. It's like you can go in much more educated and make a difference in your treatment. I mean, that's just all, to me, all up and down this supply chain. It's not just the pharmaceutical. It's not just the therapeutic, the intervention. It's so much more. And it takes education and trying to figure it out. So there's still a little ways to go, but this is where I think is it's such the fire that we're now able to do this piece.
Yeah, Mark, what are the countries that are out there that are really internationally? Who are the people that are really behind this stuff? And where is the U.S. in comparison? You know, I hear Japan is like really popping, like it's the big thing out there. So, are we like right behind them, or are we like way off?
Well, I'm actually working on a couple projects in Asia right now where we're actually looking at the precision medicine side of it. And not to make it too much about the project or my work in that specific project, but to classify it the way you're asking, seeing the more advanced annual physical examination, medical care, and then layer on top of that the technology like the not Wi-Fi, the cellular network, 5G or whatever next generation it is,
Right.
starting to employ the remote home monitoring technology. It's not just the latest AliveCore or FDA approved stuff that we have here. They're almost biohacking and having whatever case on your phone to measure X, Y, and Z. We're looking to go into some of these almost public-private partnerships for international precision medicine, so that we can do some of these, I don't want to say regulated, but more scientific controlled trials, so that we can vet and verify the gold standard of, let's say, Alzheimer's in a brain MRI, fMRI, to, let's say, an app that will measure so many different interactions and features that it can predict that you might develop Alzheimer's in the next 10 years.
Right.
So to combine some of these, it's very much Asia. And so we're looking at Korea, Singapore, Japan, and honestly, China, Taiwan, some of these other areas that, you wouldn't necessarily think are at that forefront, but there are some government, not subsidies, but, government grants and government initiative behind it. Right. And yes, you look at the U.S., there was the Obama Precision Medicine Initiative. They've changed the name on that. And you even look at Google's baseline study and things. They're not using imaging, my area, as much when you compare that to the UK and the UK Biobank. I mean, they are giving what I'm looking at, a digital physical examination, like 20 different MRI scans, German population study. It's so much more of a, I guess, cultural shift where they want to bank everything. We were talking about data earlier and looking at it in different ways. I mean, it's the time and now. That. Yes, it definitely is right down there.
Futuristic things like, gathering all that information, then having algorithms to help predict stuff is fantastic. And making an impact in areas where there's no impact is amazing. And Robin, I know that you've been involved with some organ work. Can you tell us a little bit about that?
Sure. So yeah, I co-founded a nonprofit called the Organ Preservation Alliance. We catalyze breakthroughs in tissue engineering and cryopreservation in order to eradicate the world's transplant shortage. So right now, we work a lot with the White House, the Department of Defense, and DARPA to help release both grant funding, but also provide, like the OSTP, with the education on what's going on in tissue engineering and cryopreservation, which was a big part of that $160 million initiative the White House launched last year on solving the transplant shortage. So in the world of cryopreservation, it's really exciting. It used to be about a 35-year-out problem. We thought the cryobiologists in the world kind of agreed that it was probably not going to be solved for 35 years. We've got it down to a 9-stage engineering process, essentially, where no individual one of the 9 stages needs to be more than 90% complete for cryopreservation to work. Why this is so important is because 80% of donor hearts go unused, because a heart only lives about a few hours outside the body. So you can't have a motorcycle accident in Florida and transplant that heart to somebody who's in California. Literally, the logistics don't work. So you end up throwing away about 80% of hearts. If we can cryopreserve them and create essentially an organ bank, we will be able to eradicate the wait list for hearts in something like 18 months. It's crazy. So right now, cryopreservation, we think it will be solved now in between 4 to 8 years, just by being able to aggregate the scientists and get the funding towards them and what they need. So that's what's going on in cryopreservation. In tissue engineering, do you want me to talk a little bit about what I'm seeing there?
Yeah, please.
Sure. Oh, but
Robin, I have a question. Sure. You said like we could preserve the whole heart as such and could be used within 4 to 8 years, right? So
no. Being able to cryopreserve, like that scientific problem, will be solved within 4 to 8 years. Once you cryopreserve something, it's essentially, you can keep it for a long period of time, whether it's hours or months or years, right? That's the goal, at least.
Is that like Ted Williams?
But,
can we bring Ted Williams back?
But then how are you going to regain the electrical conductivity of the heart when you retrieve the heart? Because that's an organ like that keeps functioning even after you die, I feel. right? So, I mean, to regain that electrical conductivity in the cryopreserved heart, isn't it going to be difficult?
Of course, yes. And this is, remember, we're looking at something that was probably 3 decades out to hopefully within the decade, but there are still lots of hurdles to be, go through. And when you're talking about a vascularized organ, you're talking about all of them from, kidneys and livers, hearts and lungs, right? And so they're all going to be different in how you're able to wake them up. That's part of the 9-stage engineering process. Is the last 3 stages are essentially that is unfreezing them, right? But but I can move on a little bit into what I'm seeing in tissue engineering. And remember, I'm not a scientist. I'm actually the engineer that raises the money for companies. So I'm on the business side, on the business and finance side. But what I'm seeing in like tissue engineering, of course, we can do little tiny sections like we were talking about earlier in the podcast. That's being used for drug toxicity. There's just a few cells, but for livers and kidneys, Organovo, of course, is doing that. But we're seeing out of a company called Lung Biotechnologies based on the East Coast. That's Martine Rothblatt's company. And hopefully she can be in human trials at least, or trying within hopefully a decade. What they're doing is they're taking something called ghost organs. So taking whether it's a human heart or a pig heart, right? Decellularizing them, which means wash away all of the cells, right? But leave the scaffolding in place, which is kind of like the stuff that's surrounding it, and leave potentially the vascular system in place. But we are making headway into that, so I'll get back to that in a little bit. And then repopulate the organ with the recipient patient's stem cells. And so that's a technique that Lung Biotechnologies is using. They're also looking at 3D printing the scaffolding first, and then populating it with the tissue, the recipient patient's stem cells. In terms of the vascular system, the problem is with these tiny little capillaries are extremely difficult to do, specifically because like the weight of things will actually it'll crush them, right? So Jennifer Lewis's lab over at Harvard has made a major breakthrough a past year or so in things like capillaries. So that's one to watch. So between those 3, Organovo, Lung Biotechnologies, and Jennifer Lewis's lab at Harvard, those are 3 really good things to watch in the world of tissue engineering.
That's very cool. I know that vascularization is the main thing. Does anybody else hear the echo? Oh, good. They just took care of it. I remember, I thought it was so cool when they were talking about vascularization and trying to get those capillaries. And I think somebody.
Matt, you just hit your mute button.
Sorry, they were working with a sugar factory, a confectioner's, so that because they could make this very fine trail of sugar and like a fine lattice, and then they were coating it with stem cells and then putting hot water through, which was just so cool, What are you doing here, Pat? You getting us back to the beginning here? Stem cells, progenitor cells.
Yeah, I'm just kind of toying around. You're still.
I mean, Sans could tell you all about this. She's doing this left and right. Yep. I'm so psyched that we're here with you guys, and you're actually part of this conversation that is for real. It's happening right now. You're scientists and business development people in the field. The only weird one is Pat, and he's there. He's just with his beard, But
it's just.
But really, no.
Nonstop.
It's great to have all of us here, and we're going to bring more people into the conversation. There's no doubt about it, So, all right, can I ask a little bit about CRISPR-Cas9? We hadn't really touched on gene editing that much, and I'm just trying to get through a few things. What you guys think about it might not be your area of expertise, but CRISPR, which is clustered, regularly interspaced, palindromic. Is it repeats?
Right.
No,
You're right.
That's it.
Yeah.
Okay, great. So CRISPR-Cas9 is gene editing, and right now, I mean, there's a battle between the 2 camps that said they were the first ones, and this guy, is it Zhao, I think his name is, he got in front by paying a little bit more money, and oh my gosh, it's like they're battling between Berkeley and whatever the other school is. But the point is that the reason they're battling and it's such high stakes is this technology is unbelievable. It's unbelievable. anywhere. All of our plants can change. All of our babies can change. But the thing that's got me is like Down syndrome can change for good. Parkinson's, sickle cell anemia could change for good. We can eradicate possibly AIDS, other things that can just, we're talking about CRISPR-Cas9. I'm excited that the potential is there to eradicate things like Down syndrome and Parkinson's and sickle cell anemia. And, tell me, is anybody else excited about this? Can you find any reason like this might make a difference?
That's my dream. That's my next step, that I want to work on CRISPR-Cas9 after my graduation.
Oh, nice. That's a good thing. Dreaming is a good thing. Mark, you were biting your lip there. I know. What's your story with or your experience with CRISPR-Cas9?
Well, I'm not as in depth with that aspect of it, but I was reading a story about the encoding of data. I think it just came out earlier this afternoon of the horse picture within the DNA. So basically, like steganography, right? Encoding data within there, and is that going to become your entire iPhoto database in a cell on your body, keeping all your data with you at all times? I mean, that's just that's beyond cyborg, I guess, right?
Yeah, it's next level.
Yeah. Go ahead, Pat.
Okay, let's break that off. Hold on. I'm out teching myself.
Yeah, no, I mean, there's so many concerns. Yeah, go ahead, Elad. Let's hear from you.
Yeah, I'm sorry, I missed a bit of the discussion, but I have to say that I don't know many biologists that I'm gonna run and treat themselves with any CRISPR-Cas9 therapy as yet. I think that we have tweaking our DNA is a pretty dangerous territory. And this is where I think that the limits of the patient say have to be set. It's all very well to say, oh yeah, I can eradicate this and that, because let's assume at the moment that we are right and we found one gene that causes diseases. It's a fashionable idea. Geneticists love it. It's hardly ever happened. But let's say that we can down a specific disease to a specific gene that we can edit. It became completely clear that nobody has bothered to really look how specific this is. And when we are in cancer territory, it's very easy to go to the FDA and say, here is a critical gene, I'll edit this, because ultimately, if the other option is that the patient is going to die, you can pass almost anything. This is why drug companies love cancer, because you can throw anything at cancer patients. At the end of the day, the alternative is always worse, right? But once we're talking about people with horrendous hereditary diseases that, you have to treat babies, you'll have to expose them to a manipulation that they would live with, if all is well, for the next possibly 100 years of their lives. You have to be damn sure that you're not tweaking something that you don't know that you're tweaking. And there's been a big,
but we're just talking about a patient that was going to maybe die without it. We're saying these are patients that have diseases that could actually, they're going to die with unless you try to do something. It's a little bit of a balance, and I'm sure this is what it's all about.
Yes and no, because the one study that came out, and I know there's been a lot of criticism about the methodics of it, and it may be a red herring, but it just demonstrated how little we know, has shown that there's a lot of off-target effects. And it's all very well to say, look, here is a, these are now here, the whole business with Charlie Gard has been putting mitochondria back on the map, horrendous mitochondrial mutations. You probably would not be able to help these patients because when they're born, they're already with amazing defects anyway. So, you'll have to perform it that, it's probably impossible. But let's speak about something that, you might be able to help a 3-year-old. if you're going to give them multiple cancers, they may come very early in their life. I'm not sure that the trade-off is something that the parents would look at and say, well, yeah, they'll have stronger heart, but every week we'll go to the clinic with a different kind of cancer. You know, these balances, we need to figure out how to make these things more specific. I'm not saying that we can't get there, but there is this drive now to, okay, I've heard that someone is doing this in mice or rats. It must be okay to go and put it in people. We have to sometimes, you know, I thought that what Robin was talking about, cryopreservation, I'm not sure that I believe that it's 4-8 years away, but I'd love her to be right about that. But you know, these are the scales that we're talking about years. We cannot really expect to have these things just transforming what we're doing in the clinic, even in a big hospital tomorrow, because the risks are immense. Even if you can get the money to do it, the risks to the patients are absolutely huge because we haven't figured this out.
Yeah, but you know what can you learn, right? If you don't swing the bat, you're not going to hit the ball. So
Right.
if you're at the last stages, like when you see your mother in a bed and she can't move, and it's like, you know, you're either like, please die now, or like, you know, give her an injection of whatever. I don't care. You know, like, let's see what can happen. I mean, that's what I would be. I'm thinking, oh my God, what would I want? I would just be saying, do whatever you want to do. Try to snap me out of this.
You know, this regenerative medicine is something like, you know, you drive car on the road. I mean, you do your best to drive safely, but then if you meet with an accident, I mean, that's the fate. I mean, but you do your best. Only when you drive, you'll be able to travel from one destination to the other. Unless and until you drive or face something, you'll not be able to overcome that. Right?
Yes.
I mean, I don't know. That's what I feel.
I just feel that we, at the moment, we leave a lot of it to the last stage where we actually do this in the patient. I think that we can do a lot more to look at things like risks and efficiencies, and
I agree.
that's less popular. It's less catchy. You won't be reading about this in the New York Times. It's like, you know, drug toxicity. That's what kills a lot more medicines than anything else. But you won't be hearing about all of these great drugs that failed because, oops, it killed some rat. So with all due respect to our love of lab rats and whatnot, at the end of the day, between killing a patient and killing a rat, I think most of us prefer the rat to go. You know,
no, I totally agree to that. I really want the scientific community to publish even their negative results so that, you know, we at least read them and we know that this is not going to work, and we don't keep trying them again and waste our labor, time, money, everything. So that is something which I'm very upset about the scientific community, because they never want to publish negative results.
Well, I think that I'm not sure that it's true, at least about small molecules, i.e. chemical-based approaches, because what tends to happen is that actually you may not know what the negative data is, but if you look you know, at any medical chemistry journal, what the companies always publish are the things that failed. Because if it's actually good and the thing that they're going to make money, that waits until it gets to the FDA, right? What you'll get is exactly the failures. So I think that the mechanism is there. I think that what doesn't get published enough is actually the clinical trial data. I think that there's always this headline, going to ASCO or something like that. That percentage failed, percentage, but you don't get the nitty-gritty. You don't get access to the material, so we can understand why things fail. Certain things, like you said, have to happen in the clinic, have to happen in people, because ultimately rats are not people.
But if you don't,
we have to build a platform to learn.
if you don't make that transition from the mouse, you know, to people, then where are you going to be? You know, I pulled up my man Thomas Edison again when he was, you know, he claims that I have not failed. I've just found 10,000 ways that won't work. Basically, that's how he discovered the light bulb. That was his mindset. And I believe if that comes to be more, get back in there, as it becomes a more prevalent approach to, you know, science, where, I mean, do we want our scientists to be like, oh my God, I'm gonna kill a rat, or I'm gonna kill, you know, I don't want to make the transition, and then FDA is like, well, you know, you have to pay us, and it's all bureaucratic. It's like, come on, man, take the shackles off, and let's take it to the next level. Let some of these guys get out there, try some things on some people that are, you know, that, I mean, put it this way, if I have a chronic disease and I say, hey guys, I can't live with this thing anymore. You can do whatever you want to me. And if you fail, you fail. I think a lot of people are willing to take that medicine, you know, and what happens is everybody's so handcuffed. And why are they handcuffed? I mean, I think that's one of the things that we should be talking about. Why is the FDA, why is it so hard to get things through the FDA? I can tell you that back to Organovo, and you know, when you're talking about tissue and they're doing their tissue patch and they put it inside a mouse, and I spoke to the CEO personally, and I listened to, I watched his body language, and he was so excited when he talked about the success that it's having inside mice, and they put that patch on, and what it's doing to the other cells, and it's growing, and it's kind of like catching a wave, and it's making, you know, bad cells turn into good cells. You know, I don't want to start claiming into any science stuff, but because I don't know, because this is why this is a talk about for dummies. Because
no, I think that was published, buddy, and you're right. The patch was in one place, and then the cells next to it actually started to regenerate. So yeah, you can check that. That's science. It's in the study.
And the conversation that I think is important is just that. It's like, why does Japan want to do it? Why is Japan leading, like saying, Organovo, come over here and we're gonna jumpstart your deal, and we're gonna get it kicking. We're gonna put you off to the races. If they don't want to do it over there, and then why are people flying into other places to you know, they're at last ditch effort. Hey, you know, play with my cells. Give me an injection. Do something, or I'm about to jump off the building. That's where a lot of people are. So who is it for anybody to say that a person cannot do what they want to do? You got to take the safety bars off. You got to, like, I mean, you can't put safety bars on every roof and say, oh, we're going to stop people from jumping off buildings. That's important.
All right, you're rambling. All right, no, here's the thing. I agree with you, and I feel a little bit weird because
far away, you get punched in the. Go
ahead. my brother-in-law had a stroke. He's like 55 years old. He was a guy that had 2 jobs, would sleep like 4 hours, life of the party, and an amazing guy. And he had a stroke like, I don't know, 4 years ago, something like that. And I keep looking at some of the articles that I see, potential for stem cells can help. You know, I mean, he's taking Botox to try to get more movement in the muscles, and I don't want to suggest it to him. And I think that's part of it too. You don't want to get people's hopes up. You know what I mean? Like, that's the thing. Now, if you're the person,
Matt, you could use a little Botox. I see those wrinkles. Let me get a little close up there.
Listen. I want you guys to get this. All right. My cousin Pat here is going to join me to run the Baltimore Marathon, but he's not going to do any training. The cuckoo's over your head, buddy. You know? So, no, but it's like, we don't want to give people hope. You know what I mean? Like, it's, if you kill their hope, it's even worse, you know, but it's not. I think that there's an opportunity to pursue these therapies in bioprinting for implantation in the clinic. Also, it'll help with all your, you know, studying actual human tissue rather than animals. Then you've got CRISPR-Cas9, which can help a lot of people with genetic disorders. And then you've got stem cell therapy, which the iPSC cells, maybe they're a little bit, you know, subject to change, maybe not. Let's study it more. Let's get it in Elon's
why don't we throw it around the horn?
office.
Why don't we throw it around the horn here and see what people think?
Go ahead,
Robin. You want to tell us what you think about pushing science? Is it something that is a moral thing where the government should be involved so much to the point where they can tell someone what they can take and what they can't take?
I look at it a little bit of a different way. I look at it as who's going to pay for it, right? So the government and the FDA is regulating things, which allows reimbursement to happen with insurance companies and Medicare and stuff like that. If you're talking about an untested drug, right, that might have catastrophic side effects that could cost millions of dollars in things like ICU costs or something like that, right? So I'm all for patients being able to do whatever they want with their own bodies. I'm absolutely pro on everything. If you want to take an experimental drug, all of that, absolutely. But what it comes down to is it's not their responsibility at that point. If you're going to take something so experimental and you're going to take your own health into your hands, it's not your insurance companies, the governments, or the hospital systems. These are all for-profit agencies for the most part, right? It's not their responsibility then to foot the bill for potentially a $10 million bill in an ICU if you're in there for 6 months on a ventilator, right? So I think it comes down to that for me.
I absolutely agree with you, and I'm not a proponent of having the government shell out a bunch of cash to go, hey, well, you try this and you try that and you try this. I don't think that's the case, but I mean, when someone can either afford it themselves or I mean, you're going to get to the then there's a situation where you're going to get to the poor and the rich, and the poor don't have it and the rich have it. But if the rich had computers at the very beginning, right? The rich had see, I don't know because I wasn't rich. So what do they have?
I mean, that's true with anything. Once any technology hits the scene, whether it's a VCR, a microwave, or a drug, right? It could potentially, it's not democratized until it falls in price. We're seeing that with 3D printers. We're seeing that with virtual reality now.
I mean,
right?
Betamax and VHS. I mean, they were high priced, and TV sets and things, all that stuff. But but it's your life, and you should be able to do what you want. And if you do the due diligence, and you should do the due diligence if you're going to get something injected into your body. I mean, I haven't even been to a doctor in 20 years. So for me, I stay clear of most of that, and I try to eat healthy and be healthy, but there may be a time, and I think, I know a doctor saved my life early on. I mean, if it wasn't for C. Everett Koop or Dr. Palmieri, I probably would have not been around. So I was fortunate to have that connection, and being, the proximity between Philadelphia Children's Hospital was where kind of close to where I grew up, and I was fortunate to get in to meet to have that doctor. So, and some people don't, but I just feel like you are the person that makes that final decision of what you want. And I think science would get a huge uplift from that. And I sure I'm not in the drugs, don't really like all that stuff, but if the FDA was a little bit less stringent on the stuff that they do, because I think there's a lot of bureaucracy in it, and I don't know, I'm just kind of throwing that out there, but who knows. Mark, what do you got, man? What do you think?
Well, I was going to say the government, the bureaucracy component is a little bit more of the regulation, legislation, and all of those aspects, commercialization too, right? But you look at biohacking, and maybe let's take it down a little bit from genetically reprogramming ourselves and everything there. Excuse me, and just look at combining the closing the loop for diabetes with the continuous glucose monitor and the insulin pump. Just looking at the data and the people who have had to hack them, so to speak, because they've had to get access to the proprietary data and export it and try to not just manually do all the measurements and then calculate and input it. Being able to do that from a technology play at home yourself to what is it, Gene Space here in New York and Brooklyn, and IndieBio, some of the people who are batch brewing insulin on their own in their garage, you know, it's step or two down and things like that. But there are aspects that you can do that I think you can say, well, do I get the choice to put something in my body? Do I get the choice to figure something out for myself? Kind of the empowerment, the education and whatnot. But it's an entirely different level when you take that to scale and how's it going to affect other people and things. So it's a little bit of a play in between, but those times are coming. The strides that some of these people are making in the technology aspect, because they are unshackled, so to speak, and can think entirely different than the regular pathway. It's a combination. We need to also get it from anecdotal to, okay, can it be proven and tested? It's not just, oh, well, it worked for me, but, you know, I can't reproduce it. I've only got one batch that's good.
Right,
it's the premium one.
No, I totally agree with that. I mean, you have to be careful. I mean, someone's going to say, hey, look, you know, you jump in this chair, I'm going to put this electrode on your ear, and then I'm going to have a cattle prod up your nose, and it's going to get rid of cancer, you know. But some people might be that desperate where they would try something like that. But I'm saying, you know, you move it along into sophisticated studies, and then, okay, you know, I personally think some of the things that one of the companies that I know quite well is Organovo, and I think some of the stuff that they're doing can definitely be pushed along for the benefit of patients and saving lives, and not only saving human lives, but saving, you know, animals' lives. I mean, we're just testing on these animals, and it's not really a one-to-one comparison. Hey, you know what? Can you guys speak to that? Actually, since you've done a lot, I'm sure all of you guys have done a lot of tests on animals. How does the animal model help us, and how fast and how long do you think that's going to be around? Anybody?
I personally have never worked on animals, and I've always been against working with animals. I've always worked with human cells. Yeah.
Well, that's a good point.
Comment.
Why don't you flip the conversation around and say, hey, how impactful do you think it is that you can actually print liver tissue and kidney tissue and show that, I mean, they've got 2 mass spectrometers there at Organovo, so you can create diseased tissue, one. You could use it just as straight tissue and see how a drug works on it. You can check metabolism of a drug through a human tissue and see what pathways it goes to and what it breaks down to as far as its metabolites. So this is printed tissue that is behaving very much the way you want it to behave. And how about eradicating testing on animals completely? Let's embrace bioprinted tissue for testing. And I mean, Mark mentioned it before with the microfluidics. There's such an explosion in this area. Aspect Biosystems is another one. Organovo was first to market, et cetera. But there are companies that are coming out now. They're all claiming, like, look, this is more predictive. You have a species gap when you're trying to test mouse, you know, to a human. There's obviously a problem there.
There's also there's obviously some benefits, though. There's obviously some benefits, or they wouldn't be doing it. You know, like, for instance,
Charles River Labs has a tremendous amount of resources to make sure that they keep doing the business the way they do the business, because it's extremely, extremely profitable. I mean, it's just that there's not enough. People are so, change is scary to people. Change is scary. And I think Mark alluded, go ahead.
Up to about 20% of drugs actually fail in late stage trials because they are toxic to the kidney. What Organovo and these other companies are doing with tiny little 3D printed tissue engineered kidney cells can make a massive impact. When you're talking about a cancer drug can cost $10 to $12 billion to get one to market for a big pharma, A neurodrug can cost up to $15 billion. These are according to Genentech, and Biogen gave me those figures directly. And you know, if you can shave off $1 billion, because you, or even more, because you're 20% of them aren't going to be failing anymore because you've tested it on a human kidney in early stage trials, that's a really big deal.
Yeah, and really, there's no testing right now for kidney toxicity. I mean, the liver toxicity, it's cells in a two-dimensional in a dish that they die after 2 days. So there's a lot of false positives and drugs that have gone through to fail and there's nothing in kidney. So now there's a predictive tool, and that's exciting. That means that you can screen appropriately at the right stage with the right molecule that you're going to test. Go ahead and test early on whatever, but when you get serious about a drug, that's when they can use it to really determine. And you're right, they'll save billions by sidestepping the failures. Mark's dying to say something, I think.
No, not too much at the moment. I agree with what you're saying.
Yeah, I mean, I think everybody's against the. I mean, you know, everybody's leaning towards not having the animal model kind of fade away. But it does seem like there is something to it. You know, I mean.
Well,
right? I mean, they're using pig parts in humans, right? They're doing transplants with pig parts, so it's not too far. It's a stretch, but it's not that much of a stretch. But since they're doing things that like that Mark had talked about, that Robin had talked about, that Organovo is doing and stuff like that to move the science forward, that's great. You're not going to need them as much, but you know, hopefully soon there won't be needed at all.
Well, where's PETA? That's what I want to know. You know what I mean? I know they came out and they said, hey, Organovo, this is great. Like one little, but I want to see like Angelina Jolie or whoever is the spokesperson on our podcast, right?
Yeah.
Like what's going on? We need a champion. But, oh, wait a minute, maybe PETA needs something to fight so that they can still be PETA. So let's not pretend like you could actually end animal testing.
That's right. You know? There's that bureaucratic, you know, angle again. It's like, no, you got to go through the FDA and we're going to control this and that, or you know, we got to use animal models because you know, so and so giant investor or president's linked into this or that. It's so crazy, man. Let's just get to the point where let guys, you know, like Mark and a lad who left, and Sanji and Robin. I always thought you were a scientist, but you're just a great speaker. And you know, let these guys get to work. You know, roll up the sleeves and get rocking on some innovation and some new science. That I think would be a big winner.
I think that's it, because the time for innovation and then the implementation, you know, the general research influencing clinical practice is one thing, but here talking about Organovo, and it's an organ and singular. You're not looking at systemic effects. So yes, having to go back to the animal model is something where you can see the interactions. I mean, that's one thing that I'm trying to look at with imaging. Some of the latest studies out now about the gut microbiome influencing Parkinson's. That's a brain-gut interaction. That's a mechanistic thing. But are we imaging and utilizing, you know, the difference of a genotype versus a phenotype? You know, what your genes say and what your body actually expresses, whether it's by environment or the genetic makeup. You know, the animals are valuable, I hate to say it in that way, because we can test it in a system. And until we can maybe link the Organovo organs into a system, I think this is where the organs on a chip and some of the bigger data play, the innovation and trying to model it differently. You know, it's going to take combining these different areas of the computer science side with the biology side and all the translation that goes in between. So that part, and everything has a place. You're right. So system, like when you're looking at that, I get it. You're going to have more there. But there's other areas like disease tissue where they can create it, which I don't think you can quite get the same benefit. And
I think they've done some great work with NASH, so that's coming out now. So I mean, there's places for all of the therapies to come into play. So you know what I'm thinking? I think that we should I should ask you guys if you're willing to come back another time. You know what I mean?
Well, I
Sure.
mean, does anybody have anything else that they want to add or that they're dying to talk about before we wrap it up?
No, it was a good session, and I think we
Yeah.
should have more sessions like this.
Sure.
Oh, wonderful. It's so great. It was so nice to meet you guys. And Mark, I know you're in New York City, and Pat,
Yes.
you're in New York City. So if you guys want to
Mark.
connect up, I'll get you your
Yes.
information over to Pat, so that if you want to connect, that's good. And
Sounds
Robin,
great.
anytime you're in New York City, or connect with Pat, and I guess if you're ever in New York City, Sans, then that's what we
I'm
do.
going to New York
She's in
next week.
Boston. Oh, awesome.
Yeah.
Cool. Yeah,
Well,
right on.
if you need a place to stay or like a quiet place to make some
He
phone
has
calls or
what is it,
whatever, or-
Airbnb? He's an Airbnb guy. So if you
Oh,
want to
okay.
stay there.
No. That's all done.
No, my
Oh.
sister stays there, so I'm just visiting her for a
But-
week.
Oh, that's
But you can come in and hang out in one of the rooms and get all Wi-Fi'd up and grab a computer and grab a camera or anything that you would like anytime you like. My door is always open and I'm working.
Yeah,
Thank you
18
very
hours
much.
a day.
Thank you.
Yeah. But it's been
So-
a really great experience, a great conversation, and we have to have many more of these. And you guys are great advocates of the science. And I mean, here I am. Robin was on the last cast, and you speak so intelligently and so passionate that I really thought you were a scientist until you said that you weren't. It's amazing.
I get that. People either think I'm a physician or a scientist most of the time.
You kind of
but
look
I-
like a doctor if you put that thing over your face.
There you go.
Stethoscope, and you're in.
Yeah,
I would
stethoscope.
I would cast you in a movie to be a doctor.
Oh, well, thank you.
Yeah.
No, I'm the money girl. I'm raising $250 million this year across 3 companies.
Oh, wow, that's great.
Nice.
Very nice.
Yeah.
Yeah, even last year I raised money for American Heart Association,
Nice.
but I didn't raise in millions. I just raised $2,000 for my department.
That's
Yeah.
great.
That's cool.
Congratulations.
And what do you
Hey, listen,
think? What do
we're
you
gonna-
think? What do you think on future podcasts or future live casts? What could we do better? And should I bring in some finance guys? That's what I'm going to try to do. Maybe just to get somebody in the discussion that maybe either is in the money game, and then this way it could be a different dynamic because we don't need all scientists. We have some salespeople. Then we have Matt, and then I'm a bit of a wild card. So.
I always love the tangents, so I agree.
Yeah, right?
Well, the venture capitalists or a pharma person who's funding some of the early stage stuff in Regen would be interesting.
Yeah, right.
Yeah.
Awesome.
Okay.
All right, so we'll definitely consider that, and we'll see what we can do next time.
And, yeah, you guys were upbeat and positive, and I'm really just so thankful that you joined, and thank you so much. Have a wonderful
you.
evening.
Thanks for
And
having
one
us.
thing,
Thank you, everyone. Bye.
one
Good
thing
night.
I always say to
Oh.
Pat is, "Got you last."
Oh, the other thing, guys, just as we just had this game growing up.
That
I
was
always beat him. Now he's trying to make up for all the losses.
Bye,
but
guys.
If you
Bye.
can, look it up and share the cast with on your social media and stuff like that would be helpful. But in
Sure,
terms-
please send the information out.
Yeah.
Perfect.
excellent. I'm sure Matt will end up doing that
Mark
tomorrow.
and Robin, I'll get connected with you on LinkedIn.
Okay,
Sounds
perfect.
good.
Yeah.
Awesome, guys. Be well.
Bye.
Bye. Have a good night.
Bye.